and for the kind invitation and for the generous introduction and UA to organize the meeting. Today, I'm going to talk with you about one of the research lines in my lab related to the potentiation of a class of drugs, echinocandins, against Aspergillus fumigatus. You probably are aware that medical mycology is a global challenge. You know, there are millions of skin infections, mucosal infections, and several millions of people daily at risk of fatal fungal infections, allergic disease. There are really a great number of deaths caused by fungi. And we estimate now that there are more deaths caused by
Recording in
fungi
progress.
than malaria, tuberculosis, and HIV, and probably more than breast and prostate cancer, you know. In spite of all this problem, it's a highly neglected field of knowledge because we have less than 3% of the infectious disease research budget everywhere in the world is dedicated to fungal infections. And if we look to the research and development budget, you know, we are talking less than 0.1%. Really, really very, very neglected area of knowledge, you know. However, 2 years ago, we saw some progress in the area due to the fact that the World Health Organization established priority pathogens, a fungal priority pathogens list, you know. And then there are 3 groups of fungi. They were classified in terms of critical groups, high groups, and medium groups. This is about 17 to 18 fungi, species of fungi. And the 4 most important ones, the critical group, are Cryptococcus neoformans, Candida auris, Aspergillus fumigatus, and Candida albicans. Today, I'm going to talk mostly about fumigatus and Cryptococcus neoformans. Not only it's a highly neglected area of knowledge, but also there are very few drugs available to treat fungal disease. You know, we are talking about 3 main classes of antifungal drugs. When we compare to antibacterial drugs, there are dozens of classes of drugs against bacteria, you know. And then we are talking about mainly azoles, you know, that are drugs that are important for the inhibition of ergosterol production, that is the main sterol in the cell membrane of fungi. And we are talking about echinocandins that are a class that is a class of drugs that are important for the inhibition of BET1-3-glucan synthase. And we are talking about amphotericin that is important for the disruption of the cell membrane. All these 3 classes, you know, there are other drugs that are not really minor drugs, but for example, 5-flucytosine, it's really important in terms of the control of Cryptococcus neoformans. But for example, Aspergillus' most important drugs are echinocandins, amphotericin, and azoles. Azoles are the first class, you know, the first line of treatment, while echinocandins are the second line of treatment against Aspergillus. But then resistance is on the rise. There are a lot of resistance coming in terms of azoles in Aspergillus fumigates, you know. Amphotericin is very toxic, although there are several novel derivatives of amphotericin that are less toxic. Also, liposomal formulations still is a very toxic drug. Let me talk first about Aspergillus, you know, Aspergillus. Most of Aspergillus, that's a very heterogeneous kind of disease. It's composed of several different clinical entities that are dependent on the immunological status of the host. Most of Aspergillus is due to fumigatus. We are talking about when the patients are completely immunosuppressed, we are talking about one of the most severe forms of aspergillose, that is the invasive pulmonary aspergillose, the IPA. It's a very, very severe disease, and the lethality is very high. That's a very important public health problem. We are talking about estimated number of 3 million cases a year and more than 300,000 cases of IPA each year. And we are talking about a mortality rate that is up to 90% in most susceptible populations. Just to remind you, the COVID, it's about 3% mortality, 90% is really probably is one of the highest mortality rates in the infectious disease, And as I told you, there are very few drugs treated the disease. I showed you that echinocandins and azoles are the main drugs, but azoles are fungicidal drugs, while the second-line treatment, echinocandins, are fungistatic drugs. And then we raised the following question in my lab. Is it possible to potentiate caspofungin? Remember that caspofungin is a fungistatic drug. It's possible to potentiate it, probably to transform caspofungin from a fungistatic drug into a fungicidal drug by using repurposing chemical libraries. And actually, we have been doing several screens in my lab. We have been identifying several compounds that can potentiate not only echinocandins, but also azoles. But let me show to you one of these screenings, in this screening, we took 4 libraries: the pandemic response box, the NIH clinical, and epigenetic compound libraries. And those libraries, they have about 1,300 compounds. They are enriched libraries. And then we combined 20 micromolar of this 1,300 compounds with caspofungin, about 0.25 micrograms per milliliter. That is the minimum effective concentration. And as you can see here, this is a Lamar blue showing just the metabolic activity. And you can see here that green is 100% activity. And here we see activity because it's a fungistatic drug, the fungus is still growing. But then we were able to identify several compounds that can potentiate the caspofungin activity. And then we defined them as enhancers when they can, by themselves, inhibit more than 30% inhibition. And we define them as synergizers when they have less than 30% inhibition by themselves. And actually, we are more interested in synergizers, You see that we were able to identify 12 of them, and they have almost no inhibition when they are alone. But when we combine them with caspofungin, they became really very toxic, And you see, among these 12 compounds, we decided to work with brilacidin. It has a very high MIC, but when we combine with low concentrations of the drug with caspofungin, you see that immediately there is inhibition. Why we decided to work with brilacidin? Brilacidin, just showing to you here the structure. Brilacidin is a very interesting drug. Brilacidin is a kind of semi-synthetic peptide, antimicrobial peptide. It was the result of biocomputational de novo drug design, And based on manganine, this antimicrobial peptide can capture the structural and biological properties of human defensins using fully synthetic non-peptide scaffolds and side chains. You see here the structural the structure of the drug, this is there is no amino acid, there are no amino acids, no peptides, but then it's mimicking an antimicrobial peptide. That's very, very interesting because we don't have to deal with all the problems related to antimicrobial peptides production and formulation and so on. And not only that, but brilacidin is already in phase 2 for many different kinds of disease. Innovation Pharmaceuticals in the United States is commercializing it, and it is in phase 2, phase 1, and sometimes even reaching phase 3. And there are several routes of delivery for several different diseases, COVID-19, oral mucositis, and so ulcerative colitis, and so on. And then we thought, wow, that's very nice because we have already a drug that has been passing through a lot of clinical trials, and we can really probably use it against, aspergillus. And then, as I told you, brilacidin can potentiate not only caspofungin, but also voriconazole. Here, we just have a viable conidia, and then you see that when we combine cas plus brilacidin, 0.2 or 0.5, you see here that there is a lower survival. We are talking about colony-forming units. The conidia has been incubated with these concentrations, and then we plate after 48 hours, and here we have 0% survival. It's completely fungicidal. And here we see very close to 0% survival with azoles. You see here 0.5, it's dead. There are no germination, and with azoles, they germinate, but they are mostly of them are dead. It seems to be very, very important as a potentiator of caspofungin and voriconazole. We checked the nature of the interaction, and it's a synergistic interaction between caspofungin and brilacidin, and additive interaction between voriconazole and brilacidin. Brilacidin can potentiate not only caspofungin and voriconazole against susceptible isolate, but also can bypass caspofungin and voriconazole resistance. You see here, we have 3 isolates that are resistant to caspofungin, and when we apply high concentrations of brilacidin together with caspofungin, you see that there is no growth. And the same is observed when we have mutants of voriconazole resistant mutants. We can, here's the reference strain that's susceptible to azoles, and here we have colony-forming units for increasing amounts of brilacidin. It's bypassing completely both resistance to caspofungin and resistance to voriconazole. Okay, but what about the mechanism of action, How brilacidin can potentiate caspofungin against fumigators? Then, normally antimicrobial peptides, they work by disrupting membrane potential. And then we decided to check if that was the case in fumigators by using this compound, that's a fluorophore called GBAC 4,3. Normally, when this compound is not taken up by the germlings, it's not fluorescing, but as soon as it's taken up by the fluorophore, by the germline, it starts fluorescing. And this indicates, since it's taking up, that there is a problem in the membrane potential. It's disrupted, probably, the membrane potential. And here is an example in the presence of caspofungin plus brilacidin. You see here that when we count the number of stained cells by GBAC C43, when we use caspofungin or brilacidin, we have very low numbers. But when we combine both drugs, we have very high numbers, about 8%, suggesting that one of the mechanisms of action is the disruption of the membrane potential. when both drugs are combined. You see here the black bars, they are the fungus that have been growing in the presence of glucose in minimal medium. But when we starve them for 4 hours without any carbon source, still the GBAC is taken up by the cell, suggesting that is a kind of passive transport is not involving energy. It seems that really the membrane potential is disrupted in presence of brilacidin plus caspofungin. We also decided to look for if other kinases could be involved in that by screening a collection of 58 protein kinase inhibitors. And then we were able to see 2 PKIs that synergize brilacidin. Here you see it's a metabolic activity again by using LMR blue. We have the control, brilacidin alone, it doesn't cause any inhibition. But when we combine these 2 specific inhibitors alone, they have almost no inhibition, but then only very high concentrations. But when we combine them with caspofungin, with brilacidin, you see that they are potentiating, they are synergizing brilacidin. These 2 inhibitors are inhibitors related to P21 activated kinase or STK25. Both of these kinases are members of the steroid 20 superfamily of kinase. These kinases have been shown to be involved in signaling through mitogen-activated protein kinase pathways. And then we know in Aspergillus fumigatus, there are 4 MAP kinases. There are 2 paralogs of the hog pathway. One MPKB, that's the homolog of FUS3, and MPKA, that is the homolog of MPK1 in Saccharomyces cerevisiae, that is the cell integrity pathway. When we tested these 4 mutants, we saw only delta MPKA, but you see delta MPKA is very sick, it's here, brilacidin is very susceptible, very sensitive to brilacidin, and since this guy is so sick, we decided to take inhibitors of the apical kinase, that's the PKCA. The protein kinase C in Aspergillus fumigatus is an essential gene, but some inhibitors can inhibit its activity. And then we took 2 inhibitors, calfostin C and sclerotrain, and you see that when we combine calfostin and sclerotrain with brilacidin, they are synergizing, suggesting that really brilacidin is acting through the cell wall integrity pathway because it's impacting not only MPKA, that is the distal, the most important kinase that is translocated to the nucleus, and the apical kinase, that's the PKCA. Also, we tested a collection of 25 non-essential protein phosphatase. Those are deletion mutants of fumigators. This collection was constructed in my lab, and there are about 29 catalytic subunits of protein phosphatase in fumigators. 4 of them are essential, and these 25 are not essential. We tested them, and then we identified calcineurin. But again, we had the same problem because calcineurin is very sick, the catalytic subunit, the mutant is very sick, and then we decided to take advantage of cyclosporine. And then cyclosporine is very specific inhibitor of calcineurin, and as you can see here, there is a synergism between cyclosporine and brilacidin. It seems that the cell wall integrity pathway and calcineurin are involved in the mechanism of action of brilacidin. Normally, why Aspergillus fumigatus, caspofungin is fungistatic against Aspergillus fumigatus? The main mechanism of action of caspofungin, as I told you, is the non-competitive inhibition of beta-1,3-glucan synthase. What happens is that the fungus has a reduced concentration of beta-1,3-glucan, but the fungus is able to activate calcineurin, MPKA, the cell wall integrity pathway, and then replace the beta-1,3-glucan by chitin, making stronger the cell wall. And the ability of replacing beta-1,3-glucan by chitin is the true nature of the fungoscatic nature of caspofungin. And then we decided to check if calcineurin and MPKA were affecting chitin production upon brilacidin plus caspofungin. And then there are 8 chitin synthase genes in fumigators. And this is what you see here is our RT-qPCR for the 8 chitin synthase genes. You see here in the wild type, you see that there are different contributions of this chitin synthase mRNA accumulation for the biosynthesis of chitin when you use brilacidin or caspofungin plus brilacidin. You see there is some inhibition of some of them, but then when we go to the mutants, really they are much more severely inhibited than in the wild type. Also, when we measure the exposure of chitin on the cell wall by using chalcofluorides that fluoresces, and this is an indicator of the concentration of chitin exposure on the cell wall, you see that there is less chitin in the delta MPKA or more chitin in the mutants. Really completely disarranged the composition of the chitin in the cell wall. And that probably is the main reason why the cell wall is losing the ability of holding the osmotic pressure that's coming from the cytoplasm inside of the cell. And then there is a rupture and death of the cell. We also know, we tested the toxicity of brilacidin plus caspofungin combinations in the A549 pulmonary epithelial cells. And as you can see, when we use very high concentrations of brilacidin and caspofungin, and there is no toxicity, this is, we are measuring that by using XTT. It's similar to Alamar Blue. And you see that the cells are not toxic. But the most spectacular was the fact that when we applied brilacidin plus caspofungin in a chemotherapeutic murine model of invasive pulmonary aspergillose, you see that the drug, the combination of the two drugs can almost clear completely the fungus in the lungs of the mice. Here I'm showing to you, this is a kind of model where we have depletion of macrophage and neutrophils, and here you see that when it's the positive control, and here when I apply brilacidin, there is some decrease. Brilacidin works in high concentrations, and here there is some decrease with caspofungin, but you see that it's much better, the application of both drugs at the same time. In a different model of keratitis, only by applying brilacidin, we see that brilacidin can suppress the fungal burden caused by fumigates. You see here, that's the control, that's the eye of the mouse. See, very, very nice, the eye, no disease. But then when we have the vehicle or the buffer, you see that progressively there is a contamination. The eye is completely contaminated. Fungal burden is really very high here. But then when we apply very high concentrations of brilacidin, you see that it's completely almost no fungal infections like the control. And you see that the disease severity is low, the corneal thickness is very nice, and the fungal burden is really reduced. And then this shows that brilacidin together with caspofungin or only brilacidin by itself can control the fungal infection caused by fumigators. Okay, but what about other fungi? Also, we demonstrated that brilacidin can potentiate novel, it's not a echinocandin, it's a 3-terpene drug called the brexafungerp. It's also inhibiting the beta-1,3-glucan synthase, but it's not echinocandin. But also we see that brilacidin can potentiate this brexafungerp. It's also brexafungerp. The main advantage is that normally the echinocandins are injected. You have to inject the echinocandins, and brexafungerp is an oral form. And it's commercially available against Candidíase vulvaginal, but it's not really tested or available against Aspergillus yet, But we show that it's a fungistatic drug too, and brilacidin can potentiate it. But then coming back to my question, what about other fungi? No? It's possible brilacidin can potentiate other fungi, caspofungin against other fungi. And then yes, the answer is yes. We take, for example, Candida albicans, that is 8 micromolar, the MIC. If we take like 1/4 of it, and we combine with brilacidin, you see clearly that the metabolic activity and the colony-forming units is really potentiated by brilacidin. Also can bypass resistance. When you have caspofungin resistant strains, here we are working with 1/4 of the MIC, is very variable for each strain, but you see clearly this last bar here, always caspofungin can potentiate, brilacidin can potentiate caspofungin against caspofungin resistant Candida albicans. Also, brilacidin can potentiate caspofungin against Candida auris. That's a very important fungus that is really coming like very, very fast, causing a lot of troubles in many countries around the world. You see here that caspofungin plus brilacidin, we are talking about 1/4 of the concentration again. You see that it can effectively cause inhibition when combined. Also, when we combine brilacidin with poxoconazole, we can inhibit the growth of mucorales. You see here 2 FIC experiments, showing that different species of mucorales can be really efficiently suppressed their growth by combining the 2 agents. In terms of fumigators, we think the model for how brilacidin can synergize caspofungin against fumigators is the following. Caspofungin inhibits FKS1 that encodes the single beta-1,3-glucan synthase in fumigators. And when this happens, the cell wall integrity pathway is activated, and calcineurin and the MPKA, they are activated. And as I told you, many things happen, but one of the most important hallmarks is hyperbranching and the replacement of beta-1,3-glucan by chitin. And this is the main meaning of the relative survival, the activation of compensatory pathways. And that's why caspofungin is a fungistatic drug. But then brilacidin can inhibit these compensatory pathways and can convert a fungistatic drug into a fungicidal drug. Okay, let me show to you another fungus that is also a big problem. We are talking about Cryptococcus neoformans, Cryptococcus neoformans, differently from fumigators, has a completely different cycle, It can cause mainly 2 different diseases, a pulmonary disease and the important cryptococcal meningitis. Normally it's present in the environment, like aspergillus. It's in the soil, in trees, in avian excreta, like in pigeons. And normally, when it's inhalated into the lungs, it can be latent for many decades, years in the macrophage, and then suddenly can be reactivated and cause infection, and suddenly can go to the central nervous system and has the ability to go to the meningitis, and cause meningitis. And we demonstrated that brilacidin can potentiate caspofungin. That's amazing, because we know that the beta-3 glucan synthase from Cryptococcus is susceptible to caspofungin. But then we have to use very high, non-physiological, almost non-physiological concentrations of caspofungin to inhibit the growth of Cryptococcus. But not only brilacidin is potentiating caspofungin against Cryptococcus, but also brilacidin can work in very, very low concentrations. It's completely fungicidal. And then we were very excited about it, because, as I told you in the very beginning of the presentation, there are very few drugs available against Cryptococcus, and Cryptococcus can evolve resistance very quickly. Normally, people normally use fluconazole plus 5-flucytosine, amphotericin, but always there are very quickly evolution, the resistance evolving, And it's a very serious problem in terms of many regions of the world, like in Africa with HIV patients and so and so. And then here you can see that the metabolic activity of caspofungin is very high concentration, And then when we go to viability, even with high concentrations, the viability goes down a little bit. But then when we use brilacidin, immediately you see that brilacidin by itself, it's, as I told you, is very, very toxic to the fungus, but we go to 1/4 of the MIC, and 1/4 combined with very low concentrations of caspofungin can kill 100% of the fungus. It's really very, very, very important finding in terms of chemotherapy. And then we decided also to investigate, and I'm going to tell you in the next 10 minutes, I'm going to tell you what we identified as possible mechanisms of action of brilacidin against Cryptococcus. First, brilacidin potentiates not only Cas, but also amphotericin against neoforms. That's interesting, because amphotericin is one of the drugs that is used for treatment of Cryptococcus. What about the mechanism of action? How brilacidin can inhibit Cryptococcus neoformans? And then again, we decided to take advantage of chemo screenings, chemogenomic screenings. We screen a collection of 155 transcription factors, and we identified a single transcription factor in this collection, that is the SRE1. We know from the literature that the SRE1 is very important for the regulation of genes involved in the ergosterol and sphingolipid biosynthesis. And then here, I just described to you the two pathways, and we took some mutants in these pathways. And here, midiosin is an important drug that can inhibit the first step of the pathway, that is the conversion of serine palmitoyl coenzyme 8 into this sphingosine. And this is a serine palmitoyl transferase inhibition. That's the enzyme that's inhibited. And also we had some mutants here. And that's very nice, because you see here that when we took these mutants and we measured the MIC for brilacidin, you see that they have reduced MIC when compared with the corresponding wild-type strains, suggesting that brilacidin is really impacting the ergosterol and the sphingolipid pathway in these fungus. Also, we incubate the wild-type strain of Cryptococcus in presence of increasing concentrations of brilacidin, and then we used a compound that fluoresces naturally, that is Philippine. Philippine binds to sphingolipids, to lipid rafts, ergosterol, and it's beautiful here. You see that as we increase the concentration of brilacidin, we see a completely disorganization of the sphingolipids and the lipid rafts and all the ergosterol completely disorganized, suggesting that really brilacidin is messing with the sphingolipid and ergosterol organization. Also, we see interaction, as I told you, between mediocin, that is the inhibitor of the first step of the sphingolipid biosynthesis pathway, and brilacidin, confirming that brilacidin is really affecting the ergosterol and the sphingolipid biosynthesis in Cryptococcus. We also took advantage of these 58 protein kinase inhibitors, and we screened the collection again to see if we were able to identify potentiators of brilacidin, and we were very lucky. We got 3 potentiators, and this, as you can see here, by themselves, they cause a little bit of inhibition, but when we combine them with brilacidin, you see that they inhibit much more the activity, but not only the activity by using Alamar Blue, but sorry, in this case, XTT, but also they are inhibiting the viability when we measure the colony-forming units in the presence of these 3 inhibitors. Neoformans encodes 2 casein kinase from the class 1, paralogs, CCK1, CCK1 sometimes also called YCK3, and CCK2. CCK2 is an essential gene, okay? While CCK1, YCK3, we are calling here YCK3 is not. And as you can see here, when we take 2 independent deletion strains of YCK3, they are much more susceptible to the combination of brilacidin, caspofungin, than the corresponding wild type, or then caspofungin or brilacidin by themselves. Also, when we take the 2 independent mutants, you see that colony-forming units are much more sensitive to 2 of the compounds that I showed you in the previous slide, that is here and that here. The UNCA-LM39 and the PF670462, suggesting that brilacidin is impacting casein kinase 1. But what casein kinase 1 is controlling? Casein kinase 1 is the mutants are normally hypersensitive to SDS treatment. They have increased sensitivity to osmotic stress, oxidative stress, and they are important for the regulation of phosphorylation of both MPK1 and HOG1 MAP kinase, demonstrating that CCK1 is regulating the cell integrity via the MPK1 pathway, and cell adaptation to stress via the HOG1 pathway. It seems to be conserved, the mechanism of action between Aspergillus and Cryptococcus in terms of the integrity of the cell plasma membrane and also the cell wall integrity pathway. What about toxicity? Again, by using bone marrow derived macrophage, We don't see really any toxicity in very high concentrations of brilacidin. And when we expose Cryptococcus to bone marrow-derived macrophage, and we measure the viability. Here you see that's the viability after exposure, 24 hours exposure to bone marrow. But then when we use brilacidin, or a combination of brilacidin plus caspofungin, you see that brilacidin can dramatically reduce the number of colony-forming units, suggesting that brilacidin is important. But not only for the control of cryptococcus that can infect the bone marrow-derived macrophage, but also when we apply in a murine model of cryptococcus, the infection of the lung, pulmonary infection, and you see there is a modest reduction, but significant in terms of the brilacidin. We apply about 5 milligrams per kilo of the mice, and you see that there is a reduction. If you think about a lab doing that, it's very encouraging for a pharmaceutical company to invest in terms of brilacidin for cryptococcus. I just want to finish my presentation, introducing to you people who have been participating in the work. I have a group of very talented postdocs in my lab, but they are very important. But not only very talented postdocs, but I have very nice collaborators in many parts of the world, Tamara and Jean. They are very important, helping us with cryptococcus. I'm not an expert in cryptococcus. We have been learning a lot. And also Suzanne has been providing us with many drugs, like the collection of epigenetic drugs. William de Grado was the person who invented the brilacidin, and my colleagues, David Drury, who has providing all the protein kinase inhibitors. And here, I didn't mention to you because I was a little bit afraid about time, but we have been doing a lot of screenings with the collection of Saccharomyces cerevisiae deletion libraries by using barcoding, or then haploinsufficiency libraries, or temperature-sensitive libraries, overexpression libraries. And we have been interacting with Charles Boone in Toronto and his group in the Center for Sustainable Resource Science in Breaking in Tokyo. And of course, we have all the funding from our national agencies in Brazil and international agents like the Canadian Health Institutes and the National Institutes of Allergy and Infectious Disease from NIH. Thank you very much for your attention.